electrocute me. Thanks, Jim. Wow. So I've been given the delightful task of essentially giving Science of Stem Cells 101. And because there are some of my fellow New York State Department of Health types in the audience who probably know a lot about this as well, I sort of beg their indulgence as I give sort of a really brief overview of the science of human stem cell research and the different types of stem cells that we're talking about, just to lay groundwork for Amy and Bonnie and Jim and Judy's talks to come. And so I think it's important to start with just considering what stem cells are. And these are cells that are found in almost all multicellular organisms, and they have two real key properties. They have the property or the capacity of self-renewal, and this means that these cells can undergo numerous cycles of cell division. So the cell can keep dividing and giving rise to new progeny cells while still maintaining its undifferentiated state. So while still maintaining its characteristics of a stem cell. And the fact of the matter is, you as a human being, you're made up of millions or billions of different cells which die on a regular basis and have to be replaced. So you cut yourself, those cells are dead, and they have to be replaced. And what happens is there are stem cells within your body that divide and then differentiate to give rise to the replacement cells that are necessary for you to continue as an organism. And part of that is that stem cells, in addition to their ability to self-renew, they have to have what's called potency, or the capacity to differentiate into different types of specialized cells. And this concept of potency is actually a very important one because there are different levels of potency that we talk about. So in the strictest sense, the notion of potency requires that stem cells either be totipotent or pluripotent, which means that they can give rise to any mature cell type or most of the mature cell types. But sometimes we talk about multipotent or even unipotent progenitor cells with the same language of stem cells. And what I'd just like to do here is highlight what we mean when we talk about potency and totipotent versus pluripotent versus multipotent. And so, as I mentioned, potency refers to the differentiation potential, the potential of a stem cell to differentiate into different cell types. And this just illustrates what I mean here. So when we talk about totipotent cells, really what we're talking about is we're talking about the cells that you find in the very earliest stages of embryonic development, just after a couple divisions after the sperm and the oocyte have fused. And it's a structure called the morula, and these cells, we call them totipotent because these cells literally can differentiate into any type of embryonic or extra-embryonic cell type. So such cells, each one of these cells here, each totipotent cell, can give rise to a complete, unique, viable organism. And it's when you get sort of a split damage to this morula that then breaks apart and forms multiple different totipotent cells that you get what's commonly referred to as identical twinning. So shortly after fertilization, if this totipotent morula gets damaged and then splits, you'll get the situation of identical twins. Now, usually when we're talking about embryonic stem cells, we're talking about pluripotent stem cells that are pulled from the inner cell mass of a much more developed blastocyst. And these are the stem cells that are sort of derivatives of the totipotent cells, and they can differentiate into nearly all cell types. But they're not necessarily totipotent in the fact that you can't take a pluripotent stem cell and then culture it and then implant it into a uterus and will give rise to an actual full human being. But it can give rise, we believe, to almost every cell type that's present within the body. Now these cells then undergo division and differentiation to give rise to multipotent cell types. And these are the cells that can divide and give rise to a variety of different but related cell types. So usually when we talk about multipotent cells, we're talking about cells that can divide and differentiate into closely related families of cells. For example, hemopoietic stem cells can differentiate into all of the cells that you find circulating in the blood, including the red blood cells, the white blood cells, the platelets, etc. And then finally we have the unipotent cells, which can really only produce one cell type, which is their own. And so when we talk about stem cells, we have to be aware of are we talking about pluripotent stem cells like those that are derived from fertilized embryos? Are we talking about multipotent cells like many of the adult stem cells and induced stem cell lines seem to be? Or are we just talking about unipotent cells that can give rise to a single type of differentiated cell? And so really there are three different types of stem cells that are currently of interest to researchers. I'm going to go through each of these in detail, but these are the embryonic stem cells, and these are the ones that I already mentioned are the pluripotent ones derived from the inner cell mass of a fertilized embryo. There are adult stem cells, which have been isolated from various organs and tissues, and there are these newly identified induced pluripotent stem cells in which researchers can take what are called adult somatic cells. So they can take a fibroblast from your skin, and through a variety of laboratory techniques, they can induce it to revert back to a more primitive state. So they can take a differentiated cell like a fibroblast, and through a variety of either genetic or chemical techniques, they can trick it into becoming, once again, a multipotent or even a pluripotent cell. Now, the ones that we're going to talk about a lot today are the embryonic stem cells. And most of you are probably well aware of all of the controversy about this, and Jim has already touched upon this. And there's been a lot of talk about Barack Obama's recent sort of lifting of the federal ban on embryonic stem cell research. And what had happened was in 2001, then President George W. Bush announced that federal funds could be awarded for research involving human embryonic stem cells, but only if certain criteria were met. And that included the fact that they had to be cells that were derived prior to the date of his announcement. And what that limited the use of federal research money for was 71 individual genetically diverse cell lines that had been developed at that time. But the problem that a lot of federally funded researchers faced was that the majority of those cell lines were considered unsuitable for clinical purposes. A lot of them were privately held by corporations like Geron or Advanced Cell Technologies, and of those 71, only 19 were actually publicly available. And of those, none were actually suitable for clinical applications. It's a fact that most people don't appreciate. But a lot of them had been grown, for example, in cultures where they were grown on top of what are called mouse feeder cells. So these are cells that are actually derived from mice and secrete into the culture medium a variety of growth factors that keep these human embryonic stem cell lines alive. Well, that makes them unsuitable for clinical use because there is the possibility, for example, of transfer of unknown zoonotic diseases. In addition, what researchers began to see was that after several rounds of sort of growth and division, a lot of these cell lines began to exhibit genetic instability. So even though they had originally been thought to be these cell lines that sort of can self-renew without changing over time, what began to happen was in the laboratory, they began to accumulate mutations in their genome that then made them unstable, and a lot of them began to show either senescence, that is, they start dying, or they began to start losing some of their key characteristics, such as pluripotency. Now, most recently, there have been a number of discoveries. One group led by Lanza at Advanced Cell Technologies in Cambridge, Massachusetts, found that they could actually harvest, using the same techniques that are used for pre-implantation genetic diagnosis, they could harvest some of these cells from the inner cell mass without, it was believed, actually destroying the blastocyst. So that embryo could then be implanted and develop normally. That provided one potential mechanism whereby embryonic stem cell lines could be derived without running into the challenge about the destruction of a human embryo. Now, there are some limits, however, to what these new technologies can mean. For example, when we talk about the use of human embryonic stem cell lines for clinical therapeutic purposes, one of the things that we have to wonder is, will these cell lines be subject to, say, immune rejection by the host? So if we use an embryonic stem cell line for treatment, are we going to have a problem that the host will eventually develop an immune response and thus reject the cells that are inserted? And so what a lot of people have proposed is that they will actually use a technique called therapeutic cloning, whereby they'll take the human embryonic stem cell line, they'll remove that stem cell's nucleus, and they'll put in the nucleus derived from the actual patient. So they will make that embryonic stem cell line like the original patient. But that, of course, raises a whole bunch of issues about cloning. So there's absolutely no difference between the techniques that are called therapeutic cloning and the techniques that can be used for reproductive cloning. And so a lot of researchers make a semantic distinction between therapeutic and reproductive cloning, but the fact of the matter is the techniques are exactly the same. So if we try to develop policies and legislations, which I believe Jim will talk about, that address the issue of cloning, really what we're going to do is we're going to inhibit the ability of us to use therapeutic cloning procedures to get around some of the disadvantages that embryonic stem cells raise with respect to immune rejection. Now, the other two types of stem cells that you'll often hear a lot about, particularly from conservative commentators in the media, are adult stem cells and induced pluripotent stem cells. And these are different in that they are derived from actual cells that are taken from an adult host. And by adult host, we don't mean that the person has to be 21 and a driver's license and able to drink, but what we mean is that they're living human beings. So when we talk about adult stem cells, it's a bit of a semantic misnomer, but it means that a cell that is derived from an actual person rather than an embryo or fetus. And so adult stem cells have been isolated from a number of different tissue types. And what we found is that they are mostly multipotent. So they're not pluripotent. They can't give rise to every cell type within the body, but we have identified adult stem cells from a lot of different types of lineages. For example, there are examples of adult stem cells that have been isolated from bone marrow that can give rise to all of the hemopoietic cells in the body. And so this is very useful because it sort of gets around the ethical problem of destruction of an embryo. And surprisingly, adult stem cell therapies are already in wide use. Bone marrow transplants, in many ways, are adult stem cell therapies. The disadvantage, however, is that we don't know if these adult stem cells are as potent and have the same level of self-renewal capacity as the embryonic stem cells that we've been studying. They're not as easily isolated, so it's very easy to take a blastocyst, remove the inner cell mass, and culture those cells to give rise to an embryonic stem cell line. Adult stem cells represent a very, very, very small percentage of the cells actually within the body. In order to isolate them from the bone marrow, for example, you would have to extract a huge number of cells from the bone marrow and then have the techniques and the technology to pull out from that a cell population that represents less than 0.001% of the total cells you have. And then finally, even though it sort of gets around this problem of immune rejection because you could derive an adult stem cell, say from the bone marrow of the patient that you want to treat, and then grow those up in the laboratory and inject them to treat a particular disorder, thus avoiding the problem of immune rejection. The cells are exactly the same. If you want to treat a genetic disorder, you face a problem, because if you derive those cells from the person suffering from that disorder, those cells will have the same genetic mutation. And so in order to get around that, you would then have to use genetic modification techniques such as gene transfer. And then finally, there are induced pluripotent stem cells. This is a relatively new discovery. This was first identified using cells that were isolated from mice. In 2007, two research groups in the US and Japan were actually able to take human fibroblasts, and then they were able to transfect them with 4 different genes in order to trick those skin cells into reverting back into sort of progenitor pluripotent stem cells. But there are some concerns about this technology. In order to do that, you have to transfect these cells with genes, usually using either an adenovirus or a retroviral vector, which raises questions about genetic modification again. Now, there's a brand new study that just came out a couple weeks ago that suggests that you can actually just use some biotechnology, or that you can actually use some bioengineered proteins to induce the same thing by sort of taking the same proteins that the genes you would transfect into the cell, and then culture mouse or skin fibroblasts in those proteins in order to trick them into reverting back into that pluripotent stage. But that is a really, really new discovery, and it's not really clear to us yet as to whether or not those cells will be pluripotent and also have the capacity for self-renewal. And then finally, once again, you face the same challenges that we've had before with issues about immune rejection and the need for genetic manipulation or cloning in order to make these therapies that are suitable to treat a wide variety of disorders. And with that, I will stop.
So, Sean is actually, this is a very good background to what I want to talk about. So the biggest ethical issue that has been at the forefront of the discussion about embryonic stem cell research comes from the fact that the embryo is destroyed when the stem cells are removed. And so, therefore, the first thing we need to talk about is the moral status of the human embryo. And